Good day, this is the Professor, CBMD PhD, and today's Monday, November 21st, 2022. It's 11:14 and 4 seconds, a.m. Eastern Standard Time, and today's research, biological cell imaging. So let's do it here. Imaging cells, new method enables, clear, precise, look inside. Researchers can now see the fine structure and chemical composition of a human cell with unmatched clarity and precision. Date November 17, 2022. Source: Beckman Institute for Advanced Science and Technology. As biology is one of the most exciting sciences of our time, because there has always been a divide between what we can see and what we cannot see. As the smallest functional units in our bodies, cells have long commanded the attention of researchers interested in determining what they are made of and where each element resides. And together the what and the where form an all-purpose cellular blueprint that can be used to study biology, chemistry, materials, and more. And before this study, obtaining a high-resolution copy of that blueprint ranked among the impossible. And now we can see inside cells at a much finer resolution and with significant chemical detail more easily than ever. And this work opens a range of possibilities, including a new way to examine the combined chemical and physical aspects that govern human development and disease. The researchers' work builds on prior strides in the field of chemical imaging, whereas optical microscopy uses visible light to eliminate surface-level features like color and structure. Chemical imaging uses invisible infrared light to reveal a sample's inner workings. When a cell is exposed to infrared light, its temperature rises and it expands. We know from night vision goggles that no two objects absorb infrared wavelengths in exactly the same way. Comparing a poodle to a park bench is evidence enough that warmer objects emit stronger infrared signatures than cooler ones. The same is true inside a cell, where each type of molecule absorbs infrared light at a subtly different wavelength and emits a unique chemical signature. And by examining the absorption patterns, a method called spectroscopy allows researchers to pinpoint the whereabouts of each. Unlike night vision goggles, the researchers do not analyze the absorption patterns as a color spectrum. Instead, they interpret the infrared waves with a signal detector. A minute beam fastened to the microscope on one end with a fine tip that scrapes the cell surface like the nanoscale needle of a record player. Innovations in spectroscopy over the last decade have focused on steadily increasing the strength of the initial infrared wavelengths. It is an intuitive approach because we are conditioned to think of larger signals as being better. We think the stronger the infrared signal, the higher the cell's temperature becomes, the more it expands, and the easier it will be to see. A sizable setback is hidden within this approach. As the cell expands, the motion of the signal detected becomes more exaggerated and generates noise, the so-called static that impedes accurate chemical measurements. It is like turning up the dial on a staticky radio station. The music gets louder, but so does the static. In other words, no matter how powerful the infrared signal became, the quality of the chemical imaging could not advance. We need a solution to stop the noise from increasing alongside the signal. The researchers' remedy to noisy cellular imaging works by divorcing the infrared signal from the detector's movement, allowing for amplification without the added noise. Instead of focusing their energies on the strongest possible infrared signal, the researchers began by experimenting with the smallest signal they could manage, ensuring that they could effectively implement their solution before upping the strength. Though counterintuitive, starting small allowed the researchers to honor a decade of spectroscopy research and lay critical groundwork for the future. of the field. Imagine that spectroscopy researchers were in a car, headed to the Grand Canyon. Of course, everyone would think that the faster the car moves, the faster they reach their destination. But the problem is that the car is headed east from Alabama. Increasing the hypothetical car speed, his analysis, to strengthen the infrared signal, we pulled over, looked at the map, and pointed the car in the correct direction. Now, the increased speed, the increased signal, can effectively move the field forward. The researchers' map enables high-resolution chemical and structural imaging of cells at the nanoscale, a scale 100,000 times smaller than a strand of hair. Notably, this technique is free of fluorescent labeling or dyeing molecules to increase their visibility under a microscope. Researchers were critical to the study's experimental stage. The idea itself arose not from sophisticated technology, but from the culture that supported curiosity, unconventional problem solving, and diverse perspectives. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.